Hi, my name is Alexander Gillert. I'm from the Fraunhofer Institute for Computer Graphics Research in Rostock, Germany, and this work is about monitoring plant roots in time series of Minirhizotron images. Our current understanding of plant roots is highly incomplete because they are difficult to monitor. The most common techniques include digging them out and washing them, but this destroys the plant and it's not possible to create time series of the same plant. Minirhizotrons are transparent tubes that are inserted into the ground so that roots can grow around them. Then with the camera or a scanner one can monitor the roots from within the tube without destroying the plant and thus create time series of the same plant. Current methods for Minirhizotron image analysis only consider a single image at a time and only measure a single parameter, namely total root length. And this parameter does not show how much growth or decay occurred in between two measurements. This example shows two images that were acquired 1 year apart. There are some roots that are present in both images, and there are some newly grown and decayed roots. And this is the result of our method, which can measure which roots are present in both images, represented as white pixels, and which ones are newly grown in green and which ones decayed in red. This problem is not simply about overlaying two images, because roots as well as soil can move. Instead, one has to find offset vectors for every single root in both images. Additional difficulties are that roots can change their appearance, and soil can also change visibly, for example due to the moisture content. Our method consists of two neural networks. The first one is a simple semantic segmentation network that classifies each pixel in both images as root or not root. The result is then post-processed with this skeletonization procedure to create single pixel wide structures. The second network computes how similar roots in both images are. It has the same architecture as the first one, except that it generates feature maps. The features are matched with cosine similarity with skeleton pixel locations as key points. The matches are then used to warp the first segmentation map onto the second one to create the result, which we call turnover map. No annotations are required to train the similarity network. In the first one we use contrastive learning to teach the similarity network how similar two roots are. For positive views we use the same root, augmented only with pixel-wise operations such as brightness or color jitter. And for negative views we use rotations, flips and other positions in the same image. This already works in simple cases, but for better performance we continue training in the second stage. This time on roots from different images at other points in time from the same Minirhizotron experiment. For positive views, we use the matches from the first stage. We only use annotations for the evaluation. For this we have created a custom user interface, which annotators used to correct mistakes caused by our procedure. This annotation is not perfect, as we discuss in the paper. We compare our approach with established feature matching methods, such as SIFT, and with voxelmorph, a method developed for a similar task in the medical domain. Our method shows better performance on almost every compared metric. Thank you for listening. For more details, please check out the paper.